Hey medics, welcome to Histology Slide Review Part 1. In this series, we're going to be covering histology slide identification and most frequently asked questions in a marathon exam. So in this video, we'll be covering the cell, blood, connective tissue, cartilage, bone, nervous system, lymphatic system, and cardiovascular system. So first off, we have the cell. So let's start by discussing some of these questions. In terms of cell organelles and their functions, the most frequently asked is that of the mitochondria, which is oxidative phosphorylation, heat production, calcium storage, and apoptosis. Then you have that of the Golgi vesicles, which modify, sort, and package proteins produced by ribosomes. Then you have that of the rough endoplasmic reticulum, which plays a role in protein synthesis, and smooth endoplasmic reticulum, which plays a role in synthesis of lipids, phospholipids, and steroids, and also plays a role in detoxification. So in terms of cell cycle and its checkpoints, there are 4 phases of the cell cycle. You have a G1 phase, in which the cell is basically growing and increasing in size, and there's a checkpoint here which checks for DNA damage and cell size and the energy reserve of the cell. If the cell passes this checkpoint, it goes on to what's known as the S phase. If it does not, it goes to what's known as the G0 phase. So in the S phase, it basically involves DNA replication. Then after the S phase, you have the G2 phase, which involves growth in preparation for division. There's a checkpoint here too, which checks for DNA damage and chromosomal replication. Then you have an M phase, in which now the cell will divide, and this checks for chromosome attachment to spindle. So in terms of differences between mitosis and meiosis, you have things like mitosis is a single-step process, meiosis is a two-step process, mitosis is a diploid cell, meiosis haploid cells, the daughter cells. And apart from knowing the differences between these two, you should also know the various phases of each of these. In terms of meiosis 1, meiosis 2, mitosis, anaphase, prophase, metaphase, and so on. So next we have characteristics of cells undergoing apoptosis. So this basically include chromatin aggregation, shrinkage of the cytoplasm, and condensation of the nucleus. You may also be asked to differentiate between apoptosis and necrosis, in which apoptosis is programmed cell death and is a naturally occurring physiological process. Necrosis, on the other hand, is premature cell death and it's a pathological process. So another question which may be asked in relation to this includes something like features of protein synthesizing cells. So this would be like abundant ribosomes, an extensive rough endoplasmic network, and also an extensive network of Golgi vesicles, and abundant mitochondria, and so on. So also, I guess the last thing I want to talk about is the cytoskeleton components, which you may be asked, and they're basically microtubules, microfilaments, and intermediate filaments. So moving on. Okay, almost forgot to tell you, but histological sections of the cell under light microscopy are not that common. So if you want to study this further, you'll be better off suited just studying them under an electron micrograph. So that's it. Let's move on to the next section. So this is the slide of a peripheral blood smear. It's more common in physiology than it is in histology. In terms of the steps in histology slide preparation, you're going to have fixation, which involves prevention. So this is the slide of a peripheral blood smear. It's more common in physiology than it is in histology. Having said that, let's discuss some points. So steps in histology slide preparation. You have fixation, which is done to prevent autolysis. Dehydration, which is done to remove the water content of the cell in order for it to be infiltrated by paraffin, because paraffin and water are not miscible. So the cell is placed in increasing concentrations of alcohol. Then the next step is clearing, which is done to remove the alcohol. Saline is used in most cases because it's soluble in both alcohol and paraffin. Then next step is infiltration and embedding, where the cell is placed in a melted paraffin wax and the paraffin allowed to infiltrate the tissue. Then the cell and the melted paraffin are placed in a mold and allowed to harden. The next step is trimming and sectioning. So the excess paraffin and the resulting paraffin block is trimmed off. Then the paraffin block is placed in a microtome for sectioning. Then the next step is staining, which is done to differentiate between the various cell organelles. Okay, so for blood cells and their function, so you have erythrocytes, which are the most abundant. So they have a characteristic pink staining with a central pale area due to their concavity, and their function is oxygen transport. Then you have neutrophils, which are the most abundant leukocytes. So they contain a multilobed nucleus, a pale pink cytoplasm with large and small granules. Their granules contain acid hydrolysis, which digest phagocytosed bacteria during acute inflammatory reactions. Then you have basophils, which are larger than neutrophils and smaller than eosinophils, and they contain coarse purple granules, which vary in size and shape. Their granules contain histamine, which is the culprit in hypersensitivities. You have eosinophils, which are larger than neutrophils and basophils. So they contain bilobed nucleus, large red-orange granules of uniform size, and the granules contain major basic proteins, which are released to kill bacteria that's too large to phagocytose. And then you have lymphocytes, which contain a large nucleus with a thin rim of cytoplasm, and they represent active B or T cells. Then you have monocytes, which are the largest, and they contain small granules in their cytoplasm. These granules contain lysosomal enzymes and peroxidases, and it plays a role during an inflammatory response. Then you have platelets, which are the smallest, and they play a role in blood clotting. So as for extramedullary site of hematopoiesis, you have liver and spleen. If you're also asked about the factors involved in hematopoiesis, you have things like erythropoietin and thrombopoietin. Now, this next slide is of a buccal smear. The epithelia is simple squamous epithelium because it's made of flat cells with a centrally placed round nucleus. Then it's found in lungs, in the alveoli of lungs, then in the endothelium of blood vessels, in the mesothelium, and its function is basically to facilitate rapid diffusion. So let's move on. We'll discuss the rest of the epithelial types under their various organ systems. So this is the slide of dense regular connective tissue. It contains densely packed collagen fibers oriented in the same direction. So it's found in areas requiring tensile strength, such as ligaments, tendons, and aponeurosis. For the cell types in connective tissue, you have the intrinsic cells and the extrinsic cells. The intrinsic cells include fibroblasts, which is the most common cell type seen in connective tissue, and it's responsible for the formation of fibers and ground substance. Then you have fibrocytes, which are all inactive fibroblasts. You also have adipocytes, which are fat cells, and they basically store energy in form of lipid droplets. You have undifferentiated mesenchymal cells, which are pluripotent and give rise to new cells. You have fixed macrophages, also known as histiocytes, which are derived from monocytes in the blood, and they play a role in the local defense of the body. In the extrinsic cells, they mostly play an immune role. So you have plasma cells, which are derived from B lymphocytes, and they produce antibodies. You have mast cells, which contain histamine and heparin, and are involved in inflammatory reactions, allergies, and hypersensitivities. Then you have leukocytes, which play a role in inflammatory response, and free macrophages, which are derived from fixed macrophages. So this is the slide of dense irregular connective tissue because it contains densely packed collagen fibers oriented in different directions. So dense irregular connective tissue is resistant to tearing by stretching forces from different directions, and it's found in the reticular layer of the dermis. So this slide is of loose connective tissue, and the reason is because it has thin strands of collagen fibers, it is highly cellular, and has an abundant amount of ground substance. So loose connective tissue is found in the lamina propria of gastrointestinal tract and respiratory tract. It's also found in the hypodermis of skin and stroma of glands, and it serves to cushion various organs. So in terms of composition of ground substance, it contains water and electrolyte, which are responsible for maintenance of fluid balance. It also contains structural glycoproteins, which are responsible for the adhesion of cells, and mucopolysaccharides, which are responsible for the consistency and viscosity of ground substance. This next slide is of elastic connective tissue, and the reason is that it has branching bundles of elastic fibers, a scanty amount of ground substance, and it has a reduced number of cells. And elastic connective tissue is found in areas that require elasticity apart from strength. So it's found in ligamentum noussae, which in this case is the slide on the left, and it's also found in elastic arteries, which is the slide on the right. Both these slides are of reticular connective tissue. That's because they contain reticular fibers, which are darkly staining. So reticular connective tissue is found in the liver, bone marrow, and other lymphoid organs. And for the types of collagen fibers, you have collagen type 1, which is found in fibrous supporting tissue, and it's the main collagen type in fibrocartilage. Then you have collagen type 2, which is found in hyaline cartilage, collagen type 3, which is found in reticular connective tissue, and collagen type 4 and 7, which are found in basement membrane. So both of these slides are of adipose tissue. Adipose tissue in general is made up of compactly packed adipocytes, which contain lipid droplets. So during tissue preparation, this lipid droplets is dissolved, thereby giving the cells an empty appearance. For the slide in the right, it's of white adipose tissue, and that's because it has a honeycomb appearance, and the cells have a flat peripheral nucleus, and you can also see part of an interlobar septum. For the slide on the left, it's of brown adipose tissue. That's because the nuclei is round. It's the nuclei of brown adipose tissue is round and centrally placed, but on this slide it's rather hard to tell. So as for the differences, white adipose tissue is unilocular, brown adipose tissue is multilocular. White adipose tissue has a flat peripheral nuclei, and brown adipose tissue has a round central nuclei. Also, white adipose tissue has less mitochondria compared to brown adipose tissue. So as for the function of the tissue, adipose tissue acts as an energy reservoir. That's because it stores lipid droplets, which when broken down produce energy. And it also acts as heat insulator, since it's a bad conductor of heat. So you may also be asked about hormones produced by adipose tissue, and that is adiponectin, leptin, and resistin. I personally find it easier to remember resistin because if you've ever tried to lose weight, you know it's very hard. It's like the tissue is resisting against your efforts. So resistin. In case you're asked clinicals, obviously obesity. So this slide, the slide on the right, is of mesenchyme, and that's because it contains uniformly distributed matrix, sparse collagen fibers, and undifferentiated cells. So it's highly cellular. The slide on the left, on the other hand, is mucoid tissue, which is mostly found in the umbilical cord. So you can see the walls of the umbilical vein and umbilical artery towards the upper side of the slide and on the lower right corner of that slide. So in terms of the embryonic origin, all connective tissue are mesodermal in origin. And then for function, mesenchyme is the stem cell for all adult connective tissue, while mucoid tissue serves to support the umbilical vessels, and it also represents a stage of development of adult connective tissue from mesenchyme. So this is the slide of hyaline cartilage, and the reason is it contains a highly homogeneous matrix. This is because the ground substance and the collagen fibers in hyaline cartilage have the same refractive index. Hyaline cartilage also contains chondrocytes in lacuna, which are arranged in highly isogenous groups known as nest cells, and it also has a perichondrium. So let's discuss on the organization of the perichondrium. It's similar to that of the periosteum. So it has an outer fibrous layer, which is vascular, and an inner cellular layer. So as for the areas where hyaline cartilage is found, you have tracheal rings and costal cartilage. And general features of cartilage is that it's avascular and insensitive. In case you're asked on the general function of cartilage in general, you're going to have support of soft tissues. So this is the slide of elastic cartilage. It contains elastic fibers in matrix, has a perichondrium, and closely packed chondrocytes with an eccentric nuclei. It's found in the ear pinna and external auditory meatus. Its main cell type is chondrocytes, which are found in lacuna and are responsible for the production of fibers and ground substance. If you're asked on the congenital anomaly generally associated with cartilage, you have Ehlers-Danlos syndrome and Marfan's syndrome. So these next slides are both of fibrocartilage, and that's because they are made up of densely packed collagen fibers with chondrocytes in lacuna distributed in rows between the bundles of collagen fibers. So fibrocartilage is found in intervertebral discs. It's also found in the pubic symphysis, knee menisci, and so on. So the difference between appositional and interstitial growth is that appositional growth involves an increase, or rather results in an increase in width, while interstitial growth will result in an increase in length of bone. Both these slides are of intervertebral disc. So intervertebral disc contains a central gelatinous substance known as nucleus pulposus, surrounded by a more collagenous substance known as annulus fibrosus. And intervertebral disc is made of fibrocartilage, which has no perichondrium. So I neglected to mention that in the previous slide, so you can put it among the identification points. As for the functions of intervertebral disc, it provides cushioning and also acts as a shock absorber. The type of joint it forms is a fibrocartilaginous joint, also known as a symphysis. And the other type of joint in the vertebral column is a zygapophyseal joint. In terms of embryonic origin, the annulus fibrosus is from sclerotum and nucleus pulposus is from notochord. So both these slides are of bone. The one on the right shows a longitudinal section of bone, while the one on the left shows a cross-section of bone. So bone contains a Haversian system, which contains a central Haversian canal surrounded by layers of concentric lamellae. It also has Volkmann's canal interconnecting various Haversian canals, which is better seen on the slide in the right. And it also has interstitial lamellae. You can see interstitial lamellae better in the slide on the left. And you also have osteocytes found in between the lamellae. So in terms of the lamellae system of bone, you have the circumferential lamellae, which contains an inner and outer circumferential lamellae, which line the periosteum and endosteum respectively. Then you have the interstitial lamellae, which is found interspaced between the various Haversian systems. And then you have the concentric lamellae, which forms part of the Haversian system. So for the major cell types found in bone, you have osteoblasts, which secrete the matrix component of bone, also known as osteoid. Then you have osteocytes, which maintain bone. Then you have osteoclasts, which are involved in bone resorption. As for the functional differences between organic and inorganic component of bone, the organic component of bone gives plasticity to bone, thereby allowing for remodeling according to functional demands, while inorganic component of bone gives hardness and rigidity to bone. Slide A is of spongy bone, and that's because it contains bony trabeculi, which are separated by marrow space, which contains bone marrow. Then you have the osteoblast and osteoprogenitor cells on the surface of the bony trabeculi, and these cells are either cuboidal when active or flat when inactive. Then you can also see osteocytes in lacuna, which are embedded in the bone matrix. As for the slide marked B, it's of epiphyseal growth plate. So the zones in epiphyseal growth plate, you have the zone of resting cartilage, zone of proliferation, zone of hypertrophy, zone of cartilage calcification, and zone of ossification. So the difference between endochondral and membranous ossification is that endochondral ossification involves development of bone from cartilage, and membranous ossification involves development of bone directly from mesenchyme. So endochondral ossification is important in the development of long bones. The only long bone that develops by membranous ossification is the clavicle, and membranous ossification is important in the development of flat bones. So moving on. Both these slides are of skeletal muscle. The one on the right shows a longitudinal section, and the one on the left shows a cross section. So skeletal muscles contain cross striations. They're multinucleated and they have a elongated peripheral nuclei. So in terms of the functional unit of skeletal muscle is the sarcomere, and the ultrastructural feature are features in the myofilament. So you have an M line, a Z line, an A band, a H band, an I band, T tubules. So these slides are both of smooth muscle, and smooth muscle contains a single centrally placed elongated nucleus. The cells are also elongated and spindle shaped, although you can't tell that clearly from this slide, and they contain no cross striations. So for other cells with propulsive tissue properties except muscle, you have myofibroblasts, myoepithelial cells, and myoid. So these slides are of cardiac muscles. So cardiac muscles contain intercalated discs. They show cross striations. They are branched, the fibers are branched, and they contain one or two centrally placed nuclei. The ultrastructural feature unique to cardiac muscle are dyads and intercalated discs. So for the components of intracellular muscle in these intercellular junctions, is that it contains gap junctions, which allow for the conduction of the electric impulse and also passage of small molecules. And then they contain what's known as spot desmosomes, which anchor the intermediate filaments of the cytoskeleton. And then they contain what's known as sheet desmosomes or fascia adherens, which anchor the contractile component of the cell. So this slide is over neuromuscular spindle. It's surrounded by bundles of skeletal muscle, and it's also encapsulated. So the modalities it transmits are proprioception and vibration. Neuromuscular spindle contains two types of fibers. That's intrafusal and extrafusal fibers. And it also contains two types of nerve ending. That is the primary ending, which is the annulospiral, and the secondary ending, which is the flower spray. So this is the slide of the optic nerve, and that's due to the presence of the central retinal vessels and meninges. So let's take a moment to discuss on the connective tissue coverings of nerves. So you have the epineurium, which covers the entire nerve. And then you have the perineurium, which will cover a single bundle of nerve fibers, and the endoneurium, which covers individual nerve fibers, similar to that of muscles, where you have the epimysium, perimysium, and endomysium. So this particular slide is that of the spinal cord. It contains a central canal, heat-shaped gray matter with surrounding white matter, a dorsal-median septum, and surrounding meninges. So the afferent fibers of the spinal cord are lateral spinothalamic, which is for pain and temperature. You have anterior spinothalamic for crude touch and pressure, and the dorsal columns, which are for proprioception. The efferent fibers, you have pyramidal tracts, which are the lateral and anterior corticospinal, which are motor, for motor. Then you have the extrapyramidal tract. An example is rubrospinal, which controls the muscle tone of the flexor muscles. Then you have lateral vestibulospinal, which is for equilibratory control. So let's discuss on differentiating the various segments of the spinal cord. The cervical spinal cord will have a split dorsal column, a prominent anterior horn due to supply of the upper limb, except for C1 to C3, and an abundance of white matter. The thoracic spinal cord will have slender anterior and posterior horns and a lateral horn and an abundance of white matter. The lumbar and sacral spinal cords will have prominent anterior horns due to supply of the lower limbs, a bulbous posterior horn, and a single dorsal column, with the difference being the ratio of gray matter to white matter. So these slides are both of peripheral ganglion, with the one on the right being of the sensory ganglion, also known as somatic ganglion, specifically the spinal ganglion. The one on the left is that of the motor ganglion, also known as an autonomic ganglion. So identification point is presence of satellite cells. In case you're asked on the differences between a sensory and a motor ganglion, is that sensory ganglion has pseudounipolar neurons arranged in groups. It also has well-defined satellite cells, and the nuclei is centrally placed. While motor ganglia, will have multipolar neurons which are scattered, poorly defined satellite cells, and eccentrically placed nuclei. This is the slide of the cerebral cortex. So it contains ill-defined zones, it's highly cellular, and there's also presence of meninges. So the layers from outer to inner, you have the molecular layer containing the horizontal cells of Cargill, external granular layer containing stellate cells and small pyramidal cells, external pyramidal layer containing medium-sized pyramidal cells, stellate cells, and cells of Martinotti, internal granular layer containing stellate cells, internal pyramidal layer containing large pyramidal cells, stellate cells, and cells of Martinotti, and multiform layer containing fusiform cells, stellate cells, and cells of Martinotti. So functional organization of cerebral cortex is a module, which is a vertical arrangement, and it exhibits lateral inhibition. So this is currently the most widely accepted hypothesis for cortical processing of information. Slides that of the cerebellum. It contains a cortex and a medulla. Cortex is the gray matter, medulla being the white matter. The cortex has two distinct zones. You have the outer lightly staining zone, which is the molecular layer, and the more darkly staining zone, which is the granular layer. The Purkinje layer is not that distinct in this slide. Then you have presence of foliations and meninges. So the layers of the cerebellar cortex are the molecular layer, which contains stellate and basket cells. Then you have the Purkinje layer, which contains Purkinje cells, and it's a single layer. And then you have the granular layer, which contains small granule cells, which are actually the smallest cell in the body, and Golgi type 2 cells. So as for the function, the main function of the cerebella is coordination of movement, and it has two types of fibers, mossy fibers and clumping fibers, with mossy fibers exerting a more diffuse effect. So this is the slide of the pons. Due to the presence of the fourth ventricle, basilar groove, and pontine nuclei. So let's try labeling the parts. 1 is the cerebellum, 2 is the fourth ventricle, 3 is the superior cerebellar peduncles, 4 is the middle lemniscus, 5 is the middle cerebellar peduncles, 6 are the pontine nuclei, 7 is the basilar groove, 8 are the corticospinal fibers, and 9 is locus coeruleus, which contains noradrenergic neurons. I don't think you'll be asked about that, but so this is the slide of the pons. Due to the presence of the fourth ventricle, in slide B, you got the substantia nigra and crus cerebri. So let's name the parts in slide A. So 1 is the fourth ventricle, 2 is the middle cerebellar peduncles, 3 pontine nuclei, 4 corticospinal tract, 5 dentate, and 6 the cerebellum. As for the cranial nerves found at this level, Cranial nerve number 4 all the way to 9 is found in the level of the pons. So other questions you may be asked is parts of substantia nigra. So you're going to have pars compacta and pars reticulata. Parts of crus cerebri, the fibers there, you're going to have, the frontopontine, corticonuclear, corticobulbar, occipitopontine, parietopontine, and temporopontine. Clinicals involving the pons, you have degeneration of substantia nigra in Parkinson's disease. So these are the slides of the medulla. You may take a moment to study them. The more frequently asked one is that at the level of the inferior olivary nuclei. And you may also be asked the function of the inferior olivary nucleus, which is it plays a role in the control and coordination of movements and in sensory processing and also performance of some cognitive tasks. So when you have injury to the inferior olivary nuclei, you're going to have a decreased ability to perfect highly specialized motor tasks. So this next slide is of the thalamus. It's not that common in terms of examination, but if you wish to take a moment to study it, you may do so. The slide on the right is that of the pyramidal neuron. It contains a pyramidal-shaped cell body and a prominent apical dendrite, and it's found in the cerebral cortex. The slide on the left is that of the Purkinje neuron. It contains flask-shaped cell body. Dendritic arborization is also one of the identifying factors, but it's not very prominent in this slide, and it's found in the cerebellar cortex. So these are the slides of neuroglia showing microglia and astrocytes. Oligodendrocytes and epidermal cells are not shown here. For the embryonic origin, they all come from neuroectoderm except microglia, which originates from the mesoderm. For the function, the microglia have a phagocytotic role. Oligodendrocytes play a role in myelination within the CNS. Epidermal cells line the ventricles in the brain and the central canal of spinal cord. Astrocytes play a role in the formation of blood-brain barrier, regulation of ions within the extracellular space in the CNS, and neurotransmitter uptake and release. And there are two types of astrocytes. You have fibrous astrocytes, which are predominantly found in the white matter, and protoplasmic astrocytes, which are predominantly found in the gray matter. So this is the slide of the retina. Due to presence of the pigmented layer, other well-defined cellular layers, you can also see the presence of the choroid. So let's name these layers. Layer 1 is the pigmented layer, which serves to absorb light, thus minimizing internal reflections in the eye. Layer 2 is the photoreceptor layer, so it contains rods and cones. Layer 3 is the external limiting membrane, so it's a series of junctional complexes between the molar cells and the photoreceptors. Layer 4 is the outer nuclear layer, so it contains the nuclei of rods and cones. Layer 5 is the outer plexiform layer, so it's a synapse between the axons of rods and cones and the dendrites of the bipolar neurons and processes of horizontal cells. Layer 6 is the inner nuclear layer, so it contains the cell bodies of the bipolar neurons, horizontal cells, and amacrine cells. Layer 7 is the inner plexiform layer, so it contains the synapse between the axons of the bipolar neurons with those of the multipolar neurons of the ganglia. And layer 8 is the ganglionic layer, so it contains the multipolar neurons, also known as ganglionic neurons. So in some slides you may also find a layer 9 and 10, with 9 being the nerve fiber layer, which will contain the unmyelinated nerve fibers, which are going to become myelinated by oligodendrocytes after exiting the eyeball to form the optic nerve. And layer 10 is usually the internal limiting membrane, which is made of the basal ends of the molar cells. So as for the cells in this tissue, you have the main neurons. So that's the bipolar neurons, which is found in the layer 6. Its cell bodies are found in layer 6. That's the inner nuclear layer. And then you have the ganglionic or multipolar neurons. Its cell bodies are found in layer 8, also known as the ganglionic layer. And you have photoreceptor layer, whose cell bodies are found in layer 4, also known as the outer nuclear layer. And then you have association neurons. So you have the horizontal cells, which establish contact between the different photoreceptors, and amacrine cells, which will establish contact between the ganglionic neurons. You also have molar cells, which extend from the external limiting membrane to the internal limiting membrane, and they basically support the other cells. So slide labeled A is of the eyelid. So it has thin skin on one side, which is stratified squamous keratinized epithelia, and palpebral conjunctiva on the other side, which is stratified columnar epithelia. It also contains skeletal muscle in cross-section. That's the ocularis oculi muscle. Slide labeled B is that of the cornea. So it contains stratified squamous non-keratinized epithelia on one side, and on the opposite side it contains simple squamous or cuboidal epithelia. So the layers of the cornea are it has the corneal epithelia, then the Bowman's membrane, then the stroma, then the Descemet's membrane, and then the corneal endothelium. So the cornea is transparent, and that's because it's avascular. The main contributing factor is it's avascular. So it's nourished by diffusion from aqueous humor. And the cornea also has a greater refractile power than the lens. This is the slide of the cochlea, with the one on the right specifically pointing towards the spiral organ of Corti. So the identification points for the cochlea: you have multiple cavities and membranes surrounded by bone, presence of the organ of Corti, and stria vascularis. As for the cell types, you have the hair cells, outer and inner hair cells, which play a sensory role. Then you have the outer and inner pharyngeal cells, which support the outer and inner hair cells respectively. Then you have Hensel cells and border cells, which limit the outer and inner boundary of the organ of Corti. As for the components of membranous labyrinth, you have the utricle, which gives the semicircular ducts, and the circle, which gives the cochlear ducts. This is a slide of the cochlea. It contains multiple cavities and membranes surrounded by bone. The sensory modalities are auditory and vestibular modalities. During transduction of sound, there is stimulation of the eardrum by sound waves, producing a vibration, which is then amplified by the ear ossicles and transmitted into the cochlea through the oval window. These vibrations activate the hair cells in the cochlea, producing an action potential, which is then transmitted to the spiral ganglion. So the slide marked A is that of the circumambulate papillae. So it has a sunken inverted cone shape with a flat top, which is lined by stratified squamous epithelia, and it has numerous taste buds on the lateral wall. So the slide marked B is that of the taste bud. So it contains a taste pore, and it's basically lightly stained oval-shaped body embedded in stratified squamous epithelia. So Some of the cells you'll find in the taste bud is gustatory cells, which are sensory cells. Then you have substentacular cells, which are support cells, and basal cells, which give rise to substentacular and gustatory cells. Illustrates the other types of tongue papillae. So you have the filiform papillae, foliate papillae, and fungiform papillae. They're not as frequently asked as circumflex papillae, but you may take a moment to study them. So this slide illustrates some of the encapsulated sensory receptors found within the skin. The one on the right illustrates the Meissner's corpuscle, which is a cylindrical encapsulated body found within the dermal papillae. Its axon has a zigzag course, which gives it this sort of appearance, and it's involved in two-point tactile discrimination. The slide on the left shows the Pacinian corpuscle, which has a central core containing a nerve fiber surrounded by concentric lamellae of flattened fibroblasts, thus giving it a spliced onion appearance, and its modalities are pressure and vibration. So both these slides are of the spleen. So the reason is it contains red pulp and white pulp, with the white pulp having a central arteriole. It's also encapsulated, and the red pulp contains splenic cords and sinusoids. As for the functions, it plays a role in immunity and red blood cell sequestration. So the slide marked A is that of a lymph node due to presence of the subcapsular sinus and germinal centers. The slide marked B is that of the paracortical region. The main cell types in A will be B lymphocytes and macrophages. The main cell types in B is T lymphocytes. So both these slides are of tonsils due to the presence of lymphoid nodules, that's the part labeled A in both slides. Tonsillar crypts, that's the part labeled B in slide A. And connective tissue capsule, that's the part labeled D in slide A. They also contain a stratified squamous non-keratinized epithelia. You may see a little bit of that in the slide labeled B specifically. So the slide labeled A is that of lingual tonsils. The part A is lymphoid nodules, B is the primary crypt, C is the mucus gland, and D is the connective tissue capsule. For slide labeled B is that of the pharyngeal tonsils. So A is the lymphoid nodule, B is the transverse mucosal ridge, and C is the seromucous gland. So the slide labeled A is that of Hassel's corpuscles in the thymus, and the slide labeled B is that of the islets of Langerhans in the pancreas. So you may know the difference between the two, and you should not confuse them. So the slide labeled A is that of a lymph node. It contains a thin capsule, a cortex, and a medulla. The part labeled A is the cortex, which contains lymphatic nodules, which may contain germinal centers, and its main cell type is B lymphocyte. The part labeled B is the paracortex, also known as the thymus-dependent zone, and its main cell type is T lymphocyte. The part labeled C is the medulla, which contains medullary sinuses and cords, and its main cell type is B lymphocyte. For the slide labeled A, it's that of the thymus. So it's encapsulated, it's also lobulated. It has a darkly stained cortex and a lightly stained medulla, and there is presence of Hassel's corpuscles in the medulla, although that's not seen in this particular slide. So the embryonic origin of the thymus, it originates from the third pharyngeal pouch. The lymphocytes within it come from the mesoderm, and the epithelial reticular cells come from the endoderm. Its main function is that of maturation of T lymphocytes. You may also ask on the hormones produced by the thymus. So you have thymulin, thymosin, and thymopoietin, which stimulate the proliferation and differentiation of T lymphocytes. So the cell types in the thymus, you have epithelial reticular cells, thymocytes, which are the precursors to T lymphocytes, You also have macrophages and thymic nast cells. Both these slides are of lymph nodes. If you like to, you may take a moment to study them. The slide on the right is that of a muscular artery. So it has a well-defined internal elastic lamellae and smooth muscle fibers in the tunica media. The slide on the left is that of an elastic artery, so it contains elastic fibers. It has an ill-defined internal elastic lamellae, and its tunica adventitia is well-defined and contains vasa varum. So the part labeled C is tunica adventitia, part labeled B is tunica media, and part labeled A is the tunica intima, which contains the internal elastic lamellae. So the slide on the right is that of the muscular artery. So you can see it has a well-defined tunica intima and smooth muscle fibers in tunica media. The slide on the left, the arrow is pointing specifically towards a venule. Some distance adjacent to it is an arteriole, so you may note the difference between the two. As for the functions of the endothelium, you have it facilitates diffusion of substances and also production of endothelial factors, among others. The slide on the right is that of an elastic artery, so it contains elastic fibers and has an ill-defined internal elastic lamellae. The slide on the left is that of muscular tissue, so it contains smooth muscle fibers and has a well-defined internal elastic lamellae. As for the types of capillaries, you have continuous capillaries, then sinusoidal capillaries, which are mostly found in the liver and hematopoietic tissue, and fenestrated capillaries, which are found in the kidneys. So this is the slide of the heart wall. I got it from University of Michigan Histology. So you may try labeling the part labeled A to D. So this last slide is of the conducting system of the heart. You may take a moment to study it if you wish. Otherwise, we've come to an end of this section. Thank you so much for persisting through all the way to the end. I really appreciate you. So for those studying for the exams, I wish you luck. If you have any questions, please put them in the comment section below and I'll strive to answer them as soon as I possibly can. Otherwise, goodbye and hope to see you next time.